Hi everybody, this is Luke D, and this is Mechanisms of Cancer. We're going to be talking about the cell cycle today. So let's talk about what the cell cycle is. The cell cycle is cell division. It's the process by which a cell splits and becomes two new cells. So that could be when you, for example, cut yourself and you need new cells to grow back. It can be the generation of new neurons in your brain, and it can be when you're an original cell as a baby and you become bigger. The mark of cell division is that the cells generated are identical. So genetically, that is. So your foot cell is identical to your eye cell, which is identical to your skin cell genetically. What cell division is not, or what mitotic cell division is not, is mitosis, cell growth, and anything related to sex. So mitosis is actually just division of the nuclei. Mitotic cell division is the overall process. However, mitosis is just a part of it. Cell growth is different than cell division. So if you were to take a cell and it were to divide, then the resulting cell should be about half the size of the original. If they're to grow, that's a slightly different process. And because we are talking about asexual division, we are not going to be talking about generating a sperm. We're not going to be talking about fertilizing an egg. And we're not going to be talking about the process called meiosis. So let's look at what cell division actually looks like. In this video, you can actually see mitosis occurring as well as cell division. And now let's go ahead and describe what's happening. So the first way people describe cell cycle or mitotic cell division is visually. So if you look at this image, and that image, you'll see that they're all pretty much the same. So originally, these together were called interphase. The center of the cell, the nucleus here, started to change right before a cell divided. First, you saw DNA starting to condense, then you saw the nuclear envelope disappear. You started to see the DNA line up in the middle of the cell and then divide, form new nuclei, and then the cell divided after that. So let's go ahead and look at that in slightly more detail. As I said, these three phases, though they are actually different phases because they're visually similar, are called interphase. This is called prophase, where you have DNA condensation and the nucleus dissolves. After prophase comes metaphase. And metaphase isn't shown really well here. I have another image which shows metaphase very, very well. So if you look here, you can see that the DNA is actually in the center of the cell lined up. So we call that metaphase. After metaphase comes anaphase. Anaphase is when the DNA splits or divides. After anaphase comes telophase, where the DNA starts to reform into nuclei, the nuclear membrane starts to reform, and the DNA decondenses. And like I said, after that we have something called cytokinesis, which is actually outside of mitosis. So it's another part of the cell cycle. As knowledge of the cell cycle increased, so did information of what was happening. So here we have a schematic of the same thing. So this is interphase, where you have the DNA kind of in the chromatin form, which means it's a combination of condensed around histones and kind of loosely relaxed. And what that allows is this whole thing to be compact, the DNA to be compact, and at the same time it can be read by transcription factors or polymerases so that genes can be expressed. During mitosis, we said that the DNA condensed. The nuclear membrane started to dissolve, and what we have here are mitotic organizations, microtubule organization centers, and they're going to start to move to opposite sides of the poles. Now let's talk about these guys really quickly. This is actually a double-stranded piece of DNA and its copy. So this is one double-stranded piece of DNA, and this is another double-stranded piece of DNA. So if you were to take a genome and you have one copy of a genome from your mom and one copy of a genome from your dad. And that is all the genetic information really necessary for a person. And you were to rip it up into pieces, you would end up with several double-stranded pieces of DNA. And those are called chromosomes. So our genome is divided into 23 parts. This would be one of those chromosomes. This, being its exact replica, is called its sister or sister chromatid. This is somewhere between prophase and metaphase. Here you see metaphase very clearly defined. You see the sister chromatids in the middle of the cell. They are lined up. You have microtubules attaching to each of them, and you have microtubule organization centers at the opposite sides of the cell. Anaphase comes and the chromosomes or chromatids are pulled apart. Just so you know, chromatid refers to the highly condensed form of DNA. So nothing right now can be read. No polymerase is in there, no transcription factors are binding. After those split apart, the nuclear envelope begins to begins to reform, and the DNA actually starts to decondense. So it goes back to chromatin where it can be read. Now, that was just visually what happened. The cell cycle is actually far more complicated, but it's very, very neatly divided. So what you're going to have in the beginning of the cell cycle is some kind of signal that starts it all off. So those signals are going to be like grow or don't not grow. They're going to be like there's no pressure around me. So if there's a lot of pressure, a cell is going to not want to divide. It's also going to, the cell, sense things like temperature and food to make sure conditions are optimal for division. That's really important, this kind of collecting of information right in the beginning. We also want to make sure that we generate the machinery necessary for replication, which is the next step, called synthesis, and that is copying of a 6 billion nucleotide long molecule. G2 is going to be the generation of all the machines necessary for mitosis. Because DNA condenses during mitosis, nothing is going to be able to be made during that time, so everything must be made beforehand. We talked about mitosis already, and we've said that that is not cell division, but only nuclear division. And then we have cytokinesis. Now, cytokinesis is actually one of my favorite parts. I know, it's kind of weird. But if you imagine a lasso, and you imagine wrapping that around a cell and then slowly pulling it tight, I guess you could something like this. Or if you have a cell, and you pull this tighter and tighter, you would form this kind of furrow, and that's what you see here. That will cause the two cells to pinch off, and that happens in animal cells, not in plant cells, and something actually different can happen in bacteria as well. So to review, the better version of cell cycle, of the cell cycle, or mitotic cell division, involves gap 1, which is going to be interpretation of the environment, generation of machinery necessary for synthesis or replication, generation of materials for mitosis itself, and then cytokinesis. And that wraps up the cell cycle. Have a great day, because the rest of this is really difficult and you're not going to enjoy any of it ever again. Bye.